Good morning, students. The topic for today is hybridization techniques from Unit 2. Following topics will be covered in this lecture: Introduction to hybridization and its various techniques like southern, northern and colony hybridization. First of all, we'll try to understand what is hybridization. The basic principle of hybridization analysis is that a single-stranded DNA or RNA molecule of defined sequence, that is, the probe, can base pair to the another DNA or RNA molecule that contains a complementary sequence, that is, the target, with the stability of the hybrid depending on the extent of the base pairing that occurs. The basic procedure of hybridization is shown in this figure. A single-stranded target DNA is shown bound to the membrane support in figure 1. The DNA probe labeled with a detector substance is added thereafter. The DNA probe now pairs with the complementary target DNA. After this, an X-ray film is developed and the result can be interpreted. The sequence of nucleotide in the target DNA can be identified. Now, what is blotting? Blotting is a technique used for transferring DNA or RNA onto a carrier so that they can be separated, followed by the use of gel electrophoresis. There are three types of blotting techniques that we are going to discuss: southern, northern and colony blotting. This figure shows the setup of a blotting apparatus. This diagram shows the procedure of blotting. First of all, the sample DNA is isolated and is digested with the help of restriction enzymes. The digested DNA fragments are run on an agarose gel electrophoresis. Thereafter, the DNA from the gel is blotted onto a membrane. Now, the membrane contains the DNA fragments which have been transferred from the gel. After this, a probe is added which is radio labeled and is incubated. In the last step, the hybridized DNA bands are seen on exposure to an X-ray film. This diagram also shows the procedure of blotting as we discussed in the earlier slide. Now we'll discuss the first blotting technique: southern blotting. Southern blotting technique is the first nucleic acid blotting technique that was developed in 1975 by E.M. Southern, therefore the name southern blotting. This diagram shows the procedure of southern blotting. As we can see, first of all, the genomic DNA is isolated from cells or tissues and is then digested with one or more restriction enzymes. This mixture is loaded into a well in an agarose or polyacrylamide gel and then subjected to electrophoresis. As we know that DNA is negatively charged, so it migrates towards the anode, which is a positively charged electrode. Moreover, the smaller DNA fragments move faster as compared to the larger DNA fragments. The separated DNA molecules are denatured now by exposure to a mild alkali and transferred to a nitrocellulose or nylon paper. This results in an exact replica of the pattern of DNA fragments on the gel, which is called as blotting. The DNA can be annealed to the paper on exposure to heat approximately 80 degrees Celsius. As we can see in the figure, the nitrocellulose or the nylon paper is then exposed to the labeled probes. These probes hybridize with the complementary DNA molecules on the paper. The paper, after thorough washing, is exposed to X-ray film to develop an autoradiograph. This reveals specific bands corresponding to the DNA fragments recognized by the probe. Now we move to the next technique, that is Nathan blotting. This technique is used for the specific identification of RNA molecules. The procedure is almost similar to that described for Southern blotting and is shown in this diagram. RNA molecules are subjected to electrophoresis followed by blot transfer, hybridization and autoradiography. Now we come to colony blotting. Colony blotting is also a process of hybridization for the specific identification and purification of colonies such as the bacterial clones. This technique is depicted with the help of diagram in this slide. In this slide, we can see that the desired bacteria are grown as colonies on an agar plate. Now, when a nitrocellulose filter paper is overlaid on the agar plate, the colonies get transferred. They are permanently fixed onto the paper by heat. After this, on treatment with alkali, that is NaOH, the cells lyse and the DNA gets denatured. When these DNA prints are exposed to specific probes which are radio-labeled, hybridization takes place. The hybrid complex can be localized and detected with the help of autoradiography. A similar strategy as described for bacteria just now can be used for the identification of phages and fragments of phage libraries. Thank you, students. In case of any difficulty, feel free to contact me on my mobile number. Thank you.